We are living in the era of a genomic revolution. The first step of any gene-based biological research or diagnostic test is the extraction of nucleic acids from samples. Every day, laboratories spend considerable time on sample preparation. The traditional spin column-based extraction method requires centrifugation, which can generate aerosols. It also creates significant plastic waste. And in today's ESG-conscious world, it's time to rethink how we prepare our samples. Magnetic bead-based nucleic acid extraction eliminates the need for centrifugation, reducing aerosol formation and noise, and significantly cutting down experiment time. By capturing magnetic particles bound to nucleic acids directly in solution, the method avoids clogging issues and enables high-purity extraction even from small-volume samples. This method is also compatible with automation, allowing for high-throughput processing and improved productivity. The EcoQprep Kit uses nanosilica magnetic beads designed for optimal nucleic acid purification, enabling fast and high-purity extraction. It also serves as a sustainable solution aligned with ESG values by reducing single-use plastic consumption. What makes the EcoQprep Kit different from other nucleic acid extraction kits? First, EcoQprep contains no microplastics. Conventional magnetic beads often contain polystyrene, a plastic that can pollute ecosystems and is not biodegradable. EcoQprep's magnetic beads are made from natural materials like silica and iron oxide, eliminating concerns about environmental contamination. Second, EcoQprep enables faster purification. The Q in EcoQprep stands for quick, with a magnetic strength of 80 EMU per gram, twice that of competitors. EcoQprep beads are captured faster, accelerating nucleic acid purification. Compared to polystyrene-based beads, they reduce experimental time, avoid residual particles in solution, and minimize bead loss during washing, ensuring high yields. With EcoQprep, nucleic acid extraction can be completed in just 5 minutes. That's a 70% reduction in purification time compared to spin columns, giving researchers more time for creative work. Third, EcoQprep is simple and eco-friendly. The entire extraction is done in a single tube, making the process straightforward. It also reduces single-use plastic consumption by 50% compared to spin columns. Fourth, one kit can handle large sample volumes. Traditional spin column methods require separate kits, mini, midi, or maxi, depending on sample size. With EcoQprep, just add the appropriate tube and magnetic separation rack to use the same kit for various sample volumes. Fifth, EcoQprep is compatible with the EcoQprep 96 Lite system. EcoQprep 96 Lite can automatically extract nucleic acids from up to 96 samples or 24 large volume samples. EcoQprep enables fast, high-quality nucleic acid extraction and purification from small to large sample volumes, either manually or through automation. It works with a wide range of samples, blood, cells, tissues, and can be conveniently used under a clean bench without a centrifuge. It minimizes contamination from airborne RNases and DNases, delivering reliable results, and ensures high recovery even from trace samples using magnetic beads. More accurate, time-saving, and eco-friendly. This is the next generation of nucleic acid extraction. Explore the full EcoQprep product line today.